Oh, the chapter we're on right now? So you're
Okay, tomorrow lab is, we'll go over the lab exam, and then the rest of the time is yours. You can work on unknown, you can work on food and urine one. It's your time to do some lab work. And then remember, break is at the end of class on Tuesday, so there's no lab the following week. And then the week after we get back from class, again, labs Wednesday and Friday are for your purposes, okay? So there's no more scheduled labs right now that we're doing, except working on your unknown and getting that finished up. Unknown is due the day of your final exam. I think I said the food and urine. Did I say final exam or last day of class?
ID Baxter was last day of class.
ID Baxter was last day. I think there's a due date in your instructions for the food and urine. Whatever it is, that's the due date. Okay? Any questions? Yeah, we're doing line. Yeah, we're in line 3 now. Okay, we've been talking about cell surface molecules that are important in the overall understanding of immune function. And the first thing we talked about were MHC1s and MHC2s. Which of those MHCs are associated with antigen-presenting cells only? Juice. Okay. Which type of lymphocyte is restricted to MHC interaction? D or T? T lymphocytes are MHC restricted. Okay. Some T lymphocytes, like the killer cells, are MHC1 restricted. That means that they only see antigens on cell surfaces if they are nestled in that little groove on the MHC1 receptor. Otherwise, they can't detect it. They can't react. Okay. T helpers are MHC2 restricted. So they can only see antigens in the context of that MHC2 receptor, which really means that they only interact with mainly dendritic cells, macrophages, and possibly B cells. Those are the only ones that have those MHC2s.
What does the MHC2 do to the antigen? Does it change like conformation or something with the T cell?
No, actually the T cell has a receptor that looks for both the antigen and that MHC2.
Okay, so it binds to the cell.
Yep. 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 Okay, other thing we talked about were CD receptors, and we said that the main thing that you should concentrate on was that there were these CD molecules on the surface of immune cells, and that sometimes those were kind of characteristic of the immune cell type. CD8 was associated with one immune cell type. Remember which one? T killer. Okay. And helpers and regulatory cells are CD4s. Okay. And then finally, we talked about the B cell / T cell receptor. And we're kind of using the B cell receptor as our model for receptor. And then I'll say a little bit about how T cell receptors differ. But basically, we're conceptualizing a receptor as a B cell receptor. So here's our B cell receptor right here. Okay? It's embedded in the membrane of a B lymphocyte. And the functional part of that particular receptor, kind of ignore these right here. Okay, these are other membrane proteins that are kind of associated with receptor, but functionally they don't really do anything in terms of immunity. They're kind of anchors there to position this receptor in the membrane. So we've got this receptor that has 4 polypeptide chains. What are these yellow ones designated as? Lights. Those are lights. And then there are these green ones, which are the heavies. Okay? So each receptor, B cell receptor, consists of 2 heavies and 2 lights. And then, I guess I should mention this because it's in one of the other slides, but the lights are one of 2 types of light chains. Kappa and lambda. Okay? And that's not designated here. So this This yellow one right here could be a lambda light or it could be a kappa light. But if this is a lambda light over here, can this be a kappa over here? No. Okay. These two are identical to these two. Okay. The antigen binding sites occur kind of in the little cups of the hands of that receptor. Okay. What's the valence of this receptor? How many molecules of antigen? Two. Okay. And those molecules would be identical molecules to one another, right? Okay. And what's the benefit of having a valence of greater than 1?
You can bind molecules and multiple receptors and then do it as others.
Yep. Multiple receptors then can bind to a complex molecule, and when they bind to the epitopes or antigenic determinants on that molecule, because many times those epitopes are repeated, okay, there's the same epitope on several parts of the molecule. Then when those bind, that pulls the receptors together, and that really is an initiating event in endocytosis, which actually pulls that bound molecule into the cytoplasm of the cell. Now, we said that structurally, this particular region down here at the bottom of the receptor is kind of a module that's used for all receptors. Okay? But then up here, there's a lot of variability in the amino acid sequence of these portions of the polypeptide chains. And we said that there were kind of blocks of genes that coded for these particular regions and these regions. So these regions right down here are the constant regions. Okay, so there's a block of genes for these constant regions of receptor amino acid sequence. Okay? For the light chains, there are how many blocks of genes that code for those? Remember? 2. Okay, remember what they are? DJ. Okay, so there's D blocks and there's J blocks. For the heavy chains, how many blocks of genes code for this part of the polypeptide? 3. And they are designated B, E, and J. So they have a diversity set of genes in the heavy chain. Okay? Then we talked about the fact that, okay, there's a real challenge. And that is that a particular receptor should only bind a single type of epitope or antigen. And how many epitopes or antigens do we say there are out there in the world?
Billion, trillion.
10 to the 23rd power is the estimate. A lot. Okay? And if each of these receptors was coded for by a unique gene, you would need 10 to the 23rd power genes. How many genes in the human genome? Total. Somewhere between 20 and 25,000. Nowhere near 10 to the 23rd power genes. In reality, when you start looking at the chromosomes that encode these particular receptors, there are about 200 genes total that encode receptor function. So how can 200 genes give you 10 to the 23rd gene products? So that's where we came into this concept of gene shuffling. This was really, really surprising when people figured this out, because we've always thought and conceptualized that the genome is pretty stable. But now we're finding out all kinds of things, or I don't know if Dr. Morgan McCombs talked about it in genetics, but there are things called jumping genes, where a gene can move from one location of the chromosome to another. In this particular situation, there are genes that are constantly being recombined with one another. We call that gene shuffling. And so this slide represents kind of a overview of what's going on. So let's talk about that just a little bit. What's making that noise? Okay, part of the genomic sequence in these receptor genes are some specific sets of nucleotides called RSSs. RSS stands for recombination signal sequence. Basically, those represent the boundaries between these gene blocks. Okay? So there's a set of VH genes, variable heavy genes. There's a set of DH, diversity heavy. A set of JHs, joining heavy genes. And so those sequences are kind of punctuation, if you like, in the genetic code that says this is where this block is, this is where this block is, this is where this block is. And then there's an enzyme. There's probably a whole bunch of enzymes in reality, but there's one enzyme that's been really pretty steady, pretty in depth, called RAG. And RAG stands for recombinant activator gene protein. So what this enzyme does is it starts recombining and snipping out a D here and combining it with a J. And then grabbing a V segment and joining that to a D. So now we've got this VDJ sequence. If that's combined with a C, now we've got the genetic information encoded to make a heavy chain. RAG would grab another set of these blocks, the V and J's, to make the light chain. There are multiple different types of these VHs, DHs, and JHs, or VLs and JLs. And that RAG enzyme just randomly grabs one of each of those and sticks it together to form a B-cell receptor molecule. So by chance, if you give it enough time, any combination theoretically is created of receptor structure. Now, if you look at a little bit of math, if you look at the chains here, light chains, lambda kappa, and then heavy chains, okay, how many different blocks are there for lambda chains? There are 30 of those V blocks, there are no D blocks, and there are 4 J ones, okay? So for combinations, there are 120 different combinations of VJ for kappa. For lambda, sorry. For kappa, there are 40 of these, there are 5 of these, there are 200 combinations, okay? What about heavy chains? There are 100 V blocks, there are 27 Ds, and 6 Js. And so that's 16,200 different combinations. And then when you put these together, any one of these can associate with any one of these or any one of these. So if you do all of that combinatorial math, that ends up to be about 5 times 10 to the 6 combinations. That's a lot, okay? But that's not anywhere near 10 to the 23rd power combinations. Where does that additional diversity come from? Well, one of the things that happens is that RAG enzyme randomly removes parts of V, D, and J genes before they join them together. So instead of taking the entire D3 block right here, it might snip off a few nucleotides before it grabs that block.
How does it know which part to
Good question.
cut off without
Good
it?
question. Yeah, it doesn't. It's all random. Okay? Because that's the idea, is that we form any number of these random combinations. And so we just kind of one time cuts here, one time cuts here. Yeah, it's hard to wrap your head around. I admit, it's hard to conceptualize how that works.
In that case, does it ever get just like a junk molecule that needs to be...
Oh, I'm sure it does all the time. And it gets, well, but what's a junk molecule? I mean, it's a receptor that potentially could interact with something. And so as long as you're a polypeptide chain, you've got the potential to bind to something. So you're just kind of a weird receptor, and maybe that receptor Maybe the molecule hasn't been created yet that would react with that particular receptor. That's okay. If that molecule is created, you can respond to it.
Does it ever make a receptor that would bind to human cells?
Yes, it does, and that's one of the things we're going to talk about. One of the combinations that will, by chance, result is receptor combinations that will recognize your human cells. That's absolutely true.
Is there any kind of memory system, or could like you potentially have the same type of receptor created multiple times?
You can have the same kind of receptor created multiple times. Yep. It's all random, really. Okay. I couldn't find the name of this enzyme. The source I was looking at just referred to it as another enzyme. Randomly adds nucleotides to those VDJ blocks or VJ blocks. Okay, so it decides, I'm going to stick a nucleotide in there, which will change the reading frame and make a different polypeptide chain. And the V regions, particularly, have a tendency to have high rates of mutation in those genes. It's called hypermutation. And so randomly, we can get alterations in nucleotide sequence within those gene blocks, particularly as the cell divides and forms new cells. These events really jump up the number of possible combinations now, and this is where you get ultimately that diversity of all those different receptor possibilities that could react with any potential antigen you might encounter. So what happens is that then once that receptor is put together, or I'm sorry, once the genes are put together to make the receptor, then those genes are transcribed, and then they are translated into protein. The translation occurs on the rough endoplasmic reticulum, on membrane-bound ribosomes, because these receptors then are going to be packaged up in the vesicles and inserted into the cell membrane. And this shows you a B plasma cell that has already been stimulated by its partner antigen. And we'll talk about what happens when it's stimulated in a second. But this is now in the process of starting to crank out antibody in response to that activation. And one of the things you can see here is the huge amount of endoplasmic reticulum. And that endoplasmic reticulum is going to be responsible for now secreting antibody. And what's interesting is that the structure of the antibody that's secreted is pretty much the same as the structure of the receptor that was on that cell in the first place. Okay? So they do a very similar thing when they start making antibody.
If this is like RAG does all this randomly and there's really no memory system, how does it know which combination to grab?
It doesn't.
When it's making the antibodies.
Yeah, I mean, it does know, quote unquote,
Yeah.
that it's got to take a V, it's got to take a D, it's got to take a J.
I know when it gets stimulated by its antigen and it starts making the antibodies, how does it know which VD and J to grab?
Because once you make your receptor, basically then the genome becomes stable, and it only makes that. RAG only grabs those combinations in. Okay, so it becomes fixed, basically. So that remember where this receptor stuff building is happening. That's happening during the education process in the thymus or the bone marrow. And then once you've been educated, now your genome becomes very stable, and when you go out there and sit in the lymph node, and if you get stimulated, then the signals will reactivate that enzyme to start transcribing that just one gene block that's the same gene block as the receptor. And that'll make it. Good question. Okay. John asked a very good question. Are there by chance receptors that are generated that will respond with your host cell antigens? question, answer is absolutely. So one of the things that has to be part of the education process is a phenomenon called clonal deletion. Okay? What happens is that when those cells are in the thymus, if we happen to build a cell with a receptor that will react with cell antigens, then it's going to be stimulated to undergo apoptosis and it'll die. This cell right here does not match any existing cell antigens. It's not binding to any of them like this one was. And so that cell then is okay, and it survives, and then it will be then released into the circulatory system and travel to the spleen or a lymph node or something and take up residence. And so that idea of deleting those particular cells that have self-receptors is a very important part of the education process. And that has to happen for both B cells and T cells. So clonal deletion has to happen for both B and T cells. And with T cells, it's a little bit more complicated, because what if you generate in T cells a receptor that won't recognize MHCs? Then you need to delete that cell, because those T cell receptors are supposed to be MHC restricted. So here's one that had a receptor that didn't match MHCs, so it undergoes apoptosis. Over here, here's one that reacted to self, and it's going to be deleted, and only ones that don't react to self but do recognize MHCs are allowed to survive. So this idea of clonal deletion is a really critical part. What if we get a mistake in that? What if something escapes that clonal deletion phenomenon? What would be the result? Then you have autoimmune. Okay, so here's a stem cell. Here are the progeny of this stem cell. And each one of these, because of that random shuffling of genes, has a different receptor. During the education process, some of these have been eliminated because they react to self. And so these are the surviving lymphocytes, each with a unique receptor, that go out and populate the lymph nodes or the spleen or other lymphoid tissue. Okay? So these are educated but naive. If they then are exposed to an antigen that matches their receptor, they will be activated. If the receptor doesn't match the antigen, they will not be activated. Once the cell is activated, it starts stimulated, it becomes stimulated to divide. And all those cells are clones of one another. And so basically what we're doing is we're taking this one activated lymphocyte and making millions and millions of identical activated lymphocytes. And some of these will differentiate into antibody-secreting plasma cells, and some of them will differentiate into memory cells. We're not quite ready to talk about what triggers this different differentiation pathway, but we will get there. These plasma cells will start producing antibody. This particular phenomena, where basically the antigen selects the cell that is going to be stimulated to divide and form offspring, is called clonal selection. So clonal deletion is part of the education process that deletes self-responding lymphocytes. The remainder will potentially be selected by an antigen if its receptor matches that antigen. And then the cells divide, form populations of plasma and memory cells, we start getting an antibody response. Okay? Now, What's interesting is that as these plasma cells divide, there's further mutation in the V region of that antibody gene. And as a result of these further mutations, antibodies that respond more effectively to the antigen will be selected for, and antibodies that don't respond quite as well will be selected against. So basically, during this process of cell multiplication, the lymphocytes fine-tune their response, and they get better and better at secreting antibody, which is a good fit for the antigen. So this process of kind of adapting constantly is really an important part of the immune response. Something else that's important in the immune response is cytokines. Cytokines are chemical signals that are sent from cell to cell. Typically, if the cytokines are produced by white blood cells, they're referred to as lymphokines. So the term cytokine is kind of a general term for any cell producing a signaling molecule. A lymphokine is a cytokine produced by a white blood cell. Some of these lymphokines are typically referred to as interleukins. They are chemical molecules that signal among white blood cells. So the word inter means that they are communicating, these are chemical communicating molecules between the leukocytes, the white blood cells. And they are given various designations by number. Right now, I think that they are typically numbered 1 through 35. I'm sure that the list of interleukins probably has grown since the stuff I've read has been published, but they just kind of keep adding to this list of these chemical communicating molecules. We're probably going to talk about maybe 3 of them total, those 35. We have already talked about interferons. Whoops. Remember that the gamma interferon can be produced by macrophages and lymphocytes, and this is a chemical communicating molecule. There are various growth factors which signal the bone marrow to start cranking out more white blood cells, and that can be one of your responses to infection. So your immune cells start producing these growth factors. There are various cytokines that are tumorigenic, that attach to tumor cells and cause their lysis. And then both immune cells and damaged cells can produce the chemokines. Remember that those are the molecules that are involved in chemotaxis and signaling immune cells to come to the site of infection. So the bottom line is that there are a whole bunch of chemical signaling going on during the immune response. And that makes sense if you think about it. I mean, if you're that kind of one lonely lymphocyte sitting in the lymph node that has an antigen that recognizes, a receptor that matches a particular foreign antigen, if you bind to that and start dividing, you would like to recruit other cells to come in and help you with that immune response. How do you recruit them? A lot of this chemical signaling is involved in that recruitment process. Hey, I found something problematic. Come and help. So we'll come back and talk about some of the specific chemical signaling that takes place. This particular slide just shows you some of the more common immune cytokines, what cells they're produced by, what their target is, and then what's a representative action. So for example, we will talk later about interleukin-2, one of the interleukins. And that's produced by a type of T cell called the T helper 1 cell. And its target is the T cytotoxic cell. And that provides a signal to cause the T cytotoxic cell to start dividing to form more of itself. So that's by cloning the T cytotoxic cell. Interleukin 4 is produced by T helper 2 cells. It's a different kind of helper cell. And its target is the B cell. And that causes the B cells to start differentiating into plasma cells. So when we have this branch down here of differentiation, then interleukins are going to be involved in some of that determination of which pathway of differentiation you go into. Interleukin 12 is produced by dendritic cells. Okay, here's tumor necrosis factor. Here's gamma interferon. And so these probably are the only ones that we'll really have any instance to talk about in any kind of detail at all. But please remember that the list of cytokines is much greater than this. Okay. Let's talk about antigens. Antigens are the substances that stimulate immune cells to produce antibody. They're the antibody producing or generating substances. We tend to talk about antigens I think there's some misconception probably about antigens in most people's mind, and that is when we talk about antigens, we oftentimes equate that with a foreign cell. Antigens are actually parts of molecules associated with those cells. And it's not even the whole molecule. It's a little piece of that molecule. On a complex polypeptide chain, it might be something like 8 or 10 amino acids out of several thousand in that polypeptide chain. So the specific portion of the molecule that actually stimulates the immune response is called the antigenic determinant, or epitope. So, if I have a complex protein made up of, let's say, 10,000 amino acids, and that's probably not that complex, maybe hundreds of thousands. How many epitopes would this particular molecule have? A bunch, okay? Because if each combination of unique 8 or 10 amino acids constituted an epitope, this thing is filled with these little regions that can respond to antibody. So, does one protein stimulate the formation of one antibody? No. One epitope on this protein stimulates the formation of one antibody. So if this were a flagellin protein, something associated with bacterial flagella, then flagellin doesn't just stimulate one antibody. It stimulates potentially thousands of different antibodies because that contains thousands of different antigenic determinants or epitopes. Does that make sense? So typically proteins and nucleic acids, which are pretty complex structurally, generate what we call a polyclonal response. They stimulate multiple lymphocytes, each producing their own unique antibody against that particular antigenic determinant. If only a single antibody is produced, we call that a monoclonal response. Which one's more protective? Polyclonal. I mean, think about it, okay? If flagellin on a pathogen only stimulated a single antibody, what if that particular molecule was never presented to that one cell sitting in one of the lymph nodes. It wouldn't get a response. Okay? But if it has multiple epitopes, then there's much greater chance that it will encounter one of the lymphocytes that matches one of those many epitopes. And the likelihood is that antigen-presenting cells are going to be showing all these different epitopes from flagellin to a number of different lymphocytes, and each one of those will be activated, giving us this polyclonal antibody response. There are some requirements that must be met in order for something to be an antigen. And the three basic requirements are size, shape, and complexity. Nucleic acids and proteins are pretty complex molecules. And so those typically are very good antigens. They'll stimulate a good immune response. Lipids, on the other hand, are not very complex. They tend to be made of the same repeating unit over and over again. Polysaccharides are not very complex. They tend to be made of the same unit over and over again. And so these typically elicit a pretty weak immune response. That's why we can't immunize against endotoxin, for example. That's that LPS layer molecule, and it's a lipid, and they're just horrible antigens. You inject those into an organism, and they just don't mount a good immune response. Okay, they don't have enough complexity. As far as size is concerned, typically about the minimum size is about 1,000 molecular weight. Oops. So very, very small molecules oftentimes are poor antigens. Anybody in class have an allergic reaction to costume jewelry? Yep. It's the metal in the costume jewelry, oftentimes the copper, for example. And your allergic reaction is actually an immune response. It's a T cell response. Okay? But copper is really small. How can that be an antigen? It can't. Okay? Molecules that are potentially allergenic but are too small to elicit an immune response are called haptens. And copper is a good example of a hapten. So if you inject it into the mouse in this particular case, it doesn't stimulate an immune response. But if you bind haptens to some carrier molecule, something that gives it bigger size, now it can be immunogenic. In the case of costume jewelry, what's happening is those copper ions are binding to skin proteins. And then that combination of copper hapten and skin protein is what is recognized by the body as an allergen, and that's when you get that immune response. There are several different categories in which antigens can be assigned. We've already talked about a couple of them. Autoantigens. Those are molecules that are part of yourself that elicit an immune response. And they are the basis of autoimmune disease. Typically, the autoantigen shouldn't stimulate an immune response unless there's been a failure in a process. Clonal deletion, okay? Another category of antigens are the alloantigens. Allo means other. These are antigens that are found on cells of some species, some members of this given species, but not another. Your MHC antigens are good examples, or your blood group antigens are good examples of alloantigens. And these become a real issue when we talk about transfusion and transplantation. So as long as you don't receive tissue that has an excessive amount of antigens that your body doesn't recognize, then that can be problematic in that procedure. Heterophile antigens are the same antigen found in different species. We mentioned this in the context of strep infections and rheumatic fever. There's an antigen on group A streptococcus, and that is an antigen that is also found on the cells that make up the mitral valve of the heart. And so if you have a big immune response against a strep infection, particularly one that's gone untreated, you can start producing antibodies, which then begin attacking your heart tissue.
Well, I have two questions. Why would you produce them if it's like part of the body's cells? Like, wouldn't they kill
Why would you produce?
the antigens against, or the antibodies against the heart tissue?
You're not producing antibodies against the heart tissue, you're producing them against the strep.
Right, but wouldn't, yeah.
And then that same antibody recognizes those antigenic determinants on the mitral valve cells of the heart. So it's an error.
Yeah, I guess I was just thinking that they would be like eradicated or something. Because it's like,
Yeah, I mean, typically, and I guess you're right, selection would not tend to favor this particular situation. You would like your immune response to be pretty specific and not respond to things that could also be potential foreign threats. But occasionally, it happens.
It's because they share the same binding region, right?
Yep.
so can some like foreign molecules have the same antigen?
As host cells? Yeah, they can.
No, as each
Oh,
other.
as each other? Sure. Yep. So particular two species of bacteria could share a common antigen, which would then stimulate a single type of antibody in the host.
So you can potentially like vaccinate against two different species of bacteria. With one.
I'm trying to think if there are any vaccines that are for multiple pathogens, and I can't really think of any. I don't know if that's intentional or not in the design of the vaccine. I can't think of any. This idea of heterophile antigens becomes really important in diagnostics when we start using antibodies to identify disease states, because these can give you false positive reactions. And so many times when you do your diagnostic tests, you need to set up proper controls to eliminate this possibility of false positives from heterophile antigens. Another class of antigens are allergens, and these are ones that cause an allergic response. There's a particular type of antibody that is involved in allergens that we'll talk about later on, called IgE. And people who suffer allergies seem to overproduce this particular type of antibody. We'll talk more about that later on. We mentioned we talked about pathogen superantigens. These are antigens that can stimulate T cells to produce all kinds of cytokines. We refer to that as the cytokine storm. We talked about that in the context of toxic shock syndrome.
Why is it called a storm? Is it a positive feedback system it activates? 'Cause I know.
Yeah, it just starts releasing. I mean, it's just this kind of torrent of cytokines are being released. They can cause multi-organ system failure, shock. It's a very nasty situation. And then there are classes of antigens called T-dependent and T-independent antigens. As the name implies, T-dependent antigens must have a T helper cell stimulating the B cell before antibody can be produced. So we need to have T helper plus B cell before we can get antibody. Most antigens are T-dependent. But there are some T-independent antigens that don't require the involvement of T helper cells. They can signal B cells directly to produce antibody. And we'll get into that in a little bit more detail when we talk actually about the coordinated immune response. Okay, we will talk on Monday about antibody structure. And fortunately, much of what we said about receptor structure parallels antibody structure, but there's a few wrinkles we have to throw in there. And then we're going to put this all together and see what the coordinated immune response looks like and how these various cells cooperate with one another to accomplish what we want to accomplish. Okay? So bring something with you tomorrow to work on for lab.
Okay, thank you.
In my paper, they went over MHC with the cross reactivity. In my paper, when they went